one by one. So let's start. So yesterday we were talking about enzyme activity, the kinetics of enzyme. So we had just discussed the basic SI unit of enzyme. So the international according to the international system of unit SI, it is designated as mole per second, and it is designated as Catal. It is called as Catal. KAT is the short form, right? And the concentration of enzyme activity is known to be as microcatal per liter. So it's given in microcatal per liter. So following activity units are derived from this. The specific, there is two types. There is specific catalytic activity and molar catalytic activity. Yeah, the specific catalytic activity is the activity of the enzyme preparation in relation to the protein concentration, and the molar catalytic activity, this can be determined when the pure enzyme with the known molecular weight is available. So it's expressed as a catal per mole of enzyme. So enzyme has only one active site center per molecule. The molar catalytic activity equals a turnover number, which is defined as the number of substrate molecules converted per unit time. Some enzymatic activity, so reaction rate is expressed as a change in the concentration per unit time. For enzyme catalyzed reaction, substrate turnover per unit time is commonly used. So unit is catal, mole of substrate per second. So microcatal and nanocatal are used in the medicine. So international subunit is micromole of substrate per minute. And the specific activity of enzyme, so is the enzyme activity per milligram of protein. So 1 unit would be the amount of enzyme that is converted to 1 micromole of substrate to product in 1 minute at a given pH at a given temperature. The pH should be around 6 to 8 and the optimum temperature around 25 to 37 degrees Celsius. And the specific activity is used in estimation of the enzyme purity. So, no. So turnover number, this is new term, turnover number of an enzyme. So turnover number is also termed as K catal, which is defined as the maximum number of chemical conversions of substrate molecules per second that a single site will execute for a given enzyme concentration. So K catal equals to the Vmax divided by enzyme concentration. So for example, there is a carbonic anhydrase whose turnover number is from 400,000 to 600,000 per second. That means this carbonic anhydrase enzyme molecule can produce up to 600,000 molecules of product per second. Yeah. So I will repeat, if the value is given 600,000 per second, the turnover number, that means the carbonic anhydrase enzyme has the power that can produce up to 600,000 molecules of products per second. So how to calculate this? So an enzyme has 1.84 gram molecular weight is the given weight, and the molecular weight is 36,800. So in presence of excess substrate catalyzes at the rate of 4.2 mole substrate per minute. So we have to calculate the turnover number. So first of all, we need to calculate the what do you say enzyme concentration. In order to do so, mole enzyme, what we do is 1.84 gram divided by 36,800 gram per mole equals to 5 multiplied by 10 raised to power minus 5 mole enzyme. And then to calculate the turnover number, what you do is Vmax by ET. The Vmax is given as 4.2 mole substrate per minute, and we have calculated the enzyme concentration, that is 5 multiplied by 10 raised to power minus 5. So the value would be 84,000 per minute. So 84,000 of substrate will be converted to product in per minute. So that's the turnover number of this enzyme. Same way, the catalase, carbonic anhydrase, acetylcholinesterase, chymotrypsin, lysozyme, according to their functions, they have different turnover numbers and km values. So what does K catal means? It means is a first order rate constant describing, also known as a turnover number because it describes the number of reactions that a molecule of enzyme can catalyze per second under optimal conditions. So most enzymes have K Catal values between 10 raised to power 2 and 10 raised to power 3 per second. For simple reactions, K2, K Catal for multi-step reactions and rate limiting step. So enzyme plus substrate give rise to enzyme substrate complex and then enzyme plus product. Yeah. So enzyme substrate, they form a complex. We can see there is a graph between your product and time. So different substrate have different products at different values. So enzyme plus substrate give rise to K1. Then if it goes back reversible, it is K2. If substrate goes further, then K3 enzyme plus product. So hyperbolic saturation. So this is the kinetics of relation between your substrate concentration and velocity of reaction. So reaction rate on the y-axis and substrate concentration on the x-axis, right? So it moves from there 0 till top. So what is happening is it goes up, 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 and till here it then started to go saturation point. Substrate concentration you keep increasing, the reaction rate will not increase. So the half speed where it goes, the half Vmax, it will be equivalent to the Michaelis-Menten constant. And when it's reached the top, it will be your Vmax. And remember, this substrate concentration will never reach to the Vmax point. It will be somewhere down. So how we define this Michaelis-Menten equation? So V naught is equal to Vmax multiplied by substrate divided by Km plus substrate. So V naught is equal to velocity of reaction. Vmax is the maximum velocity of reaction. S is the substrate concentration and Km is a Michaelis constant. So that's the Michaelis-Menten equation. So Km and Vmax, Km is the ratio of rate constant, that is K2 plus K3 divided by K1. From here, K2 plus K3 divided by K1. So it's equivalent to the substrate when the initial rate is equivalent to the half Vmax, and it's a property of ES complex and does not depend on the concentration of E or S. Substrate when the initial rate is equivalent. Yeah. And it's a property that ES complex does not depend on the concentration of E or S. And the Vmax is the maximum velocity at a fixed enzyme and is directly proportional to the enzyme. So the problem of Vmax in Michaelis-Menten kinetics is that it will never reach to the Vmax. So this is linear Michaelis, 1875 to 1949, and he's a Maud Menten from 1879 to 1960. So Vmax is approached but never achieved. Then comes your Lineweaver work double reciprocal plot. So they did what they did. This reciprocal of the Michaelis-Menten equation in order to identify them over the graph. So V naught is equal to Vmax Vmax multiplied by substrate divided by Km plus S. Then you have 1 by V naught equals to Km plus S divided by Vmax multiplied by substrate. So then you separate this Vmax multiplied by substrate into different parts, 1 by V naught, Km by Vmax multiplied by substrate, then S by Vmax multiplied by substrate. Then you simplify it. So this is your double reciprocal or Lineweaver-Burke equation. So S will cut out S from here. Then this will be your, it's very simple and in a nutshell shown by this Dean-Burke and hence Lineweaver both. So this equation resembles the straight line, that is y is equal to mx plus c, where y would be 1 by v naught, Your m would be your slope, that is Km by Vmax, and 1 by S would be your x-axis, and 1 by Vmax will be your intercept, that is C. This you might have done in +2 in your mathematics, that the line equation is always represented as equal to y is equal to mxc. So we have found out the slope, we have found out the intercept, and we know the y-axis, we know the x-axis. So anything that you want to calculate then, whether slope or whether the Vmax value, whether the anything, so it could be easily calculated with the help of this equation over the graph. So this is the how graph looks like of a Line-Burk a double reciprocal plot. So on the y-axis you have 1 by V naught, on the x-axis you have 1 by S naught, yeah, and on the slope it's Km by Vmax, and on the intercept is your 1 by Vmax. So it defines that equation very easily now. So this line, that is slope, that is going here, if it is goes to the negative plot of the substrate, so this intercept value would be minus 1 by Km. Yeah, if this intercept value is 1 by Vmax, but this intercept value of this x-axis, it will be minus 1 by Km. So for the same data set now, Km and Vmax can be obtained accurately as intercept values. So we can identify these Km and Vmax values if we know the velocity, if we know the substrate concentration, and we can calculate with the help of the Km, then everything is possible to calculate. Then comes the next part, that is inhibition. This is also one of the important part in enzyme engineering. We taught always, that is why we should study enzyme inhibition of enzyme activity. So there are two reasons to do this. So we can use inhibition as a probe for understanding the kinetics and properties of enzymes in their uninhibited state. So many, perhaps most drugs are inhibitors of specific enzymes. The concept of inhibition. So an enzyme is a biological catalyst that alters the rate of reaction without itself becoming altered by its participation in the reaction. So the ability of enzyme to catalyze a reaction can be altered by binding small molecules to it. So sometimes at its active site, sometimes at its site distance from the active site. So they are inhibitors and accelerators. So usually these alteration involves reduction in the enzyme ability to accelerate the reaction, which is less common, but alteration that give rise to an increase in enzyme ability to accelerate the reaction. These are known as accelerator. So why more inhibitors are available than accelerators? Because of the natural selection. If there were small molecules that can facilitate the enzyme propensity to speed up the reactions, nature probability would have been way to incorporate those facilitated in the enzymes. And most enzymes are fairly close to the optimal in their properties. We can readily mess them up with the factors, but more challenge is to make enzyme better at their jobs. So there are types of inhibitors, that is irreversible and reversible. The irreversible are the one which binds without possibility of release, usually covalently attached, and each inhibition event effectively removes a molecule of enzyme from availability. And the reversibles are attached by the weak non-covalent interactions. They are of several kinds, and classification somewhat superseded by the detailed structures based knowledge of mechanisms, but not entirely. So there are types of reversible inhibitions. They are competitive, non-competitive, and uncompetitive, and also the fourth one, mixed. So these are the all four different types of reversible inhibition. So competitive inhibition, they are the one which binds at the active site and prevent binding of the substrate. So they don't let the substrate to attach it, and it's compete the active site of the enzyme. So non-competitive, they bind a little bit to the distance from the active site, not to the active site, but little bit different site from the active site, and it's interface with the turnover number, that is the number of product being produced via substrate, so it decreases that thing. Uncompetitive, which is quite rare, it's bind to the enzyme substrate complex. So once your enzyme is there, substrate is there, they got attached, but then this uncompetitive attach at that part. So this removes ES complex and interface with the turnover number. And there is a mix, the fourth one, that's a competitive and non-competitive both. Now just remember, first of all these four parts: competitive, non-competitive, uncompetitive, and mixed. Read them again, because if you can understand these basic principles behind them, only then you can understand it further. Because we're going to talk about now kinetics of the same. Just, I will give you 2 minutes. Just read it again, please. Okay. So, how to distinguish between reversible and irreversible? So, when we dialyze an enzyme inhibitor complex against a buffer-free inhibitor, if turnover number or binding still not improve, it's irreversible. And also there is a change between competitive and other reversible sites. This kinetics and structural studies are possible. Then comes your competitive inhibition. So they are the one which increase your substrate concentration. Rate will increase as the inhibitors will be outcompeted by the increased numbers of substrate molecules. So substrate will come attached there on the side, and the inhibitor will also be coming on the substrate side. So it will be competing the same side. So let's talk about now kinetics of competition. So competitive inhibitor hinders the binding of substrate but not the reaction velocity. So the reaction velocity that is happening, it will be not hinder anymore, but mainly the binding. So it will affect the Michaelis constant but not the Vmax. So the Km is the amount of substrate that needs to be present to run the reaction velocity up to its half saturation velocity. So competitive inhibitors requires us to show more substrate into the reaction in order to achieve that half maximum velocity. So competitive inhibitor means they increases the Km. So here in this competitive inhibitor we can see Km goes up, so minus 1 Km moves towards the origin. So this is your 1 by V naught, this is your 1 by S, and this one, the first line that you're seeing is the uninhibited enzyme. So we didn't add an enzyme into that in this one. So it touches to this Vmax here, half Vmax, and doesn't have any inhibitor into that. And this in this competitive reaction, as we said, Vmax is not changed, so it will always remain constant. But your Km will be changed. So Km is minus here, minus 1 Km1. But if you add a low concentration of a competitive inhibitor, so the slope goes up, and minus 1 km it goes near to the x-axis. And same goes if you add high concentration, it goes even more close to that. And in the non-competitive inhibition, as we suggested, it will not attach to the substrate side but somewhere another side. So it does not affect the Km, but it reduces the Vmax, so it has no influence on the Km. It's the opposite way. So here in this case, in the non-competitive, your Vmax will be decreased, so your 1 by Vmax will be increased. So x-intercept is unaffected. So your slope will change. So this is uninhibited enzyme. This is your 1 by Vmax uninhibited, and this is minus 1 by Km. So this is not affected in this case. This remain constant. But once you added a low concentration of enzyme, so you will see that with the low concentration, your Vmax is increased. Yeah, but then with the higher concentration, your Vmax is even more increased. So, but your Km remains unaffected. Just think about it. I will put these things just in order to absorb these figures. So what I said, that in this case, in the competitive inhibitor, your Km is affected, your Vmax is not affected. So as you keep increasing the concentration, your Km will go up. So minus 1 by Km will move towards the origin. And in this case, your Km will remain constant, but your Vmax will keep decreasing. If your Vmax is decreasing, then 1 by Vmax will keep on increasing. So that's why the graph will go, the slope will go up and up and up. Yeah. Just think about it for 1 minute, what I said. Try to absorb these things, and then we go further. Okay, now we take the next part, that is uncompetitive inhibition. So as we talked earlier, they usually bind to the enzyme substrate complex, and it's create a ternary ESI complex. It's created really a new complex known as ESI complex, and this removes ES. So by Lee-Chatelier principle, it actually drives the original reaction to the right, so it decreases the Km. But it interferes with the turnover, so Vmax goes down. If Km and Vmax decreases at the same rate, then it's classical uncompetitive inhibition. So in this, Km moves towards the origin as you keep increasing the Vmax, and Vmax moves away from the origin, and your Vmax will moves away from the region. So both things are happening here. Neither your Vmax is constant, neither your Km is constant. Only the slope is not changing. So in earlier cases, this is your slope. This one. So it was changing frequently. This was changing frequently, but this time it will, in the uncompetitive one, it will be parallel. All three things are parallel. So first one is the uninhibited enzyme here, but once you add the low concentration, then this is uncompetitive. Yeah, if you keep increasing the concentration, it goes even more. So Vmax keep on increasing, and your -1 km will also keep increasing. So the three reversible inhibitors: competitive, non-competitive, and uncompetitive. Yeah. So this is the summary of that. I hope this one is clear now, that why we see these three different kinetics, why Km is constant in one case, why in one case your Vmax is constant, and why in one case your Km and Vmax are not constant but your slope is constant. It's just because of the enzyme, your inhibitor, where it is attaching. So in the first case, in the competitive, your inhibitor is attaching to the substrate side. In the non-competitive, it is attaching to the another side, little bit of different from it. In uncompetitive, it's attaching to the enzyme substrate complex, both together. If we have this as a subjective question, sometimes draw all these figures of inhibitors from the kinetics perspective. Then there is the allosteric enzyme. These are the ones which have other shapes and the conformation induced by the binding modulators. So these enzymes have two receptor sites. So mostly your substrate will bound to the active site, and the inhibitor will bound to the allosteric site. So inhibitor will bound to the allosteric site. So allosteric enzymes are those having other shapes or conformations induced by the binding of modulators. So these enzymes have two receptor sites. So one site fits the substrate like the other enzymes. The other site fits an inhibitor or activator molecule, and allosteric enzymes are very important in the feedback regulation. Then allosteric modulation of enzyme. So there is allosteric activation. So you have your enzyme here, allosteric site, and there is a active site. So in the activator, if there is allosteric activators are there, they will bind to the highly active site of the enzyme, and then your substrate could be attached here. These are activators and will produce lot of products. And the normal case, if there is no activator, then it will produce normal amount of products. But if there is inhibitor, allosteric inhibitor attached to the this affinity form, low activity form of enzyme, so it will produce no products. So there is allosteric activation and there is allosteric inhibition. Yeah, just try to absorb this figure. What is it saying? There is allosteric activation leading to the production of lot of products. There is allosteric inhibition leading to the little or no product formation. Or in the center, there is a lot of very few products are being produced. Now let's talk about the kinetics of allosteric enzyme. So in an earlier case, we were seeing this other curve like this very nice curve, but in this case, we see a sigmoid curve. Instead of other way, in this case, we see the sigmoid curve. So allosteric enzymes can be regulated between very low and very high reaction rates with only small changes in the substrate concentration. So allosteric enzymes are used by the cells to regulate metabolic pathways where the concentration of cellular substrates fluctuate over the narrow concentration range. So this one, instead of hyperbolic, we get a sigmoid curve. So allosteric, they show a relationship between V naught and S that differ in the normal Michaelis-Menten behavior, and they exhibit saturation with the substrate when the substrate is sufficiently high. When V naught is plotted against a sigmoid, saturation curve results, and the symbol S 0.05 or K 0.05 is represent the substrate concentration, giving half maximum velocity. So the end product, the allosteric inhibition. So you have your allosteric site with the end product here. An active site, the substrate will be attached. Enzyme is there. So substrate cannot bind. So enzyme reaction is inhibited. So enzyme reaction further in this case is proceeded. So that's it from the kinetics of all the different. I hope it was knowledgeable for all of you. So enzyme kinetics have been shared. Nomenclature and classification was shared yesterday. And then enzyme structure and mechanism yesterday we did. And we had this introduction to enzyme also. So in nutshell, so yeah, we had quite done with the topics now. Now it's time for the protein isolation. Yeah, protein isolation and purification. So this is the next topic. We will continue now. So in this topic we will discuss now about the enzyme actually, because your protein or enzyme, they are equivalent. So we will understand how they are identified, how we purify them, how we quantify them, how we use them. So some of the famous Nobel Prize winners in the case of chromatography and electrophoresis technique: Martin, Richard, Arne, yeah, from 52, 48, who worked on the partition chromatography. Then mass spectrometry and sedimentation technique by Fenn, Tanaka, and Swedberg, who provide the various big work in their field. Then the organelle fractionation in order to separate various particles from your cell. So Christiane de Duve, Albert de Claude, George E. Palade. So they did their own work. Then Frederick Sanger, who got twice the Nobel Prize winner in protein sequencing of insulin and a nucleic acid, identification of nucleic acid, in 58 and 1980. Then protein isolation, how you isolate these enzymes and proteins. First step is to disrupt them, and then root separation of homogenates into the fractionations. So homogenizations, so Downs homogenizers, sonication, osmotic shocks, French press. For the fractionation, you go for ammonium sulfate precipitation, differential centrifugation. So basically you have a homogenate in the tube, and then you centrifuge for 500 G for 10 minutes. So in the pallet you will have the nuclear fractions. Then you again with the supranatant, you take the supranatant, the top one, and then you centrifugate for the 10,000 G for 20 minutes. Then you will see that there is a pallet with the mitochondrial fractionation. And at 100,000 G for 1 hour, you will have a microsomal fraction. So they are protein isolation where you have more dense particle and less dense particle. Then you can centrifuge them, you have a pallet, and then you separate the supranatant and pallet part. Or you have a gradient. In the gradient you add mixture of small and large particles, and then you centrifuge them, and then particles settle according to the mass, and then you stop the centrifuge, and then you will have this increasing mass of particles. So these are the various centrifugation sets, that is at 3000 G for 5 minutes, 10,000 G for 10 minutes, 40,000 G for 30 minutes, 200,000 G for 16 hours, 400,000 G for 24 hours. So all these pallets will have various organelles that you can separate, like large organelle, mitochondria, membranes, large protein complexes, proteins like 50,000 Dalton. And sedimentation velocity technique, here you have low density solution, high density solution. So you have a centrifuge tube with a density gradient, then the layering of the sample is done with a rotor, and then you can separate between different from the different parts there. So this will not go. And how to quantitate this protein? So there were a Lowry method for protein determination, which could determine from 10 microgram to 1000 microgram per ml, but that was not so efficient one. Then came out the Bradford protein quantification determination. So this technique is simpler, faster than the Lowry method, and is subject to less interference. Yeah, so we use Commercy dye in this, in order which binds to the organelle and lysyl residues of proteins, and this specificity can lead to variation in the response of the assay to different proteins. So let's see how this Bradford assay is done.
In this video, you will learn how to perform protein quantitation using the Bradford assay. Protein quantitation is one of the most commonly performed procedures in a biotechnology laboratory. In this activity, the protein concentration in milk will be measured using the Bradford assay. For this activity, you will need 1x Bradford reagent and 1x phosphate buffered saline, or PBS. Using a 20 to 200 microliter micropipette, set the pipette to 98 microliters. Pipette 98 microliters of 1x PBS into an empty microcentrifuge tube labeled sample. Switch to a 2 to 20 microliter micropipette and set the pipette to 2 microliters. Pipette 2 microliters of milk sample into the microtube labeled Sample A. Mix the milk sample and PBS by either pipetting or vortexing. This mixture has diluted the milk to 1 part in 50. Label 2 cuvettes, one control and the other sample. Do not handle the cuvette where the light passes through and make certain your labels are well above this area. Pipette 20 microliters of diluted milk sample into the cuvette labeled sample and 20 microliters of 1x PBS into the cuvette labeled control. Next, we will set up protein standards. See your protocol for the correct concentrations. Label a cuvette for each standard and mark it with the corresponding concentration. Next, add 20 microliters of each protein standard into the corresponding cuvette. Make certain you use a clean pipette tip for each sample. Now you are ready to add the Bradford reagent to all of the cuvettes you have prepared, including the sample and control. Add 1 milliliter of 1x Bradford reagent to each of the cuvettes. Mix completely by pipetting up and down with the micropipette. Incubate the cuvettes at room temperature for 5 minutes. After 5 minutes, visually compare the cuvette containing the milk sample to the cuvettes containing the protein standards. Determine the standard that most closely matches the color of the milk sample. Estimate the protein concentration of the milk sample based upon the visual comparison. To determine the protein concentration using a spectrophotometer, select for the Bradford assay. When asked to insert blank into the spectrophotometer, insert the cuvette labeled control. Use the blank to set the spectrophotometer to 0 absorbance, or 100% transmittance. If instructed, read the absorbance of the 7 protein standards and record the absorbance values. Remove the cuvette labeled blank and insert the milk sample into the spectrophotometer. Read the absorbance and record the value.
Yeah, so that's the basic principle behind your in the quantitation of your chromatography. Oh, protein. There's also another method, that is ultra-sensitive fluorescence based protein quantitation, in which you add a dye to your sample, heat it for 10 minutes, make it to the cool temperature, and then you can read your fluorescence. So we get this graph like this, fluorescence versus protein, microgram per ml, and if you calculate the fluorescence value, you can tell the concentration of your protein based on that. And then protein purification dialysis. So this is a technique used to remove small molecules from the protein solution or change the buffer composition of the proteins. So you have this dialysis bag which has this big molecules, red, and small molecules. So you have this concentrated solution and you have this buffer. So at the start of dialysis, your all molecules will move out of it, and the small molecules will move out of it. Mainly your large molecules will remain in this buffer. So let's see how this dialysis is done. Okay. So now comes your chromatography part, that is in which we refer to as there is a mobile phase and solid phase, and the mobile phase is liquid, and we have a stationary phase which may be liquid or solid. So in this we have a separation of the first case of the chromatograph phase, your gel filtration chromatography, where we separate the proteins according to their size. So when you add a mixture of proteins or enzymes, and in this you will have a carbohydrate polymer beads in which your small molecules will get attached, the red ones, and the green ones will flow and the yellow ones will also flow out. So when you put into this one, they will start flowing. The yellow one will go fast, then green, and then the red. So that's the basic principle behind it. So let's see how this chromatographic is done.
We're starting with gel filtration medium. This is Cefidex G50. The number here gives you an approximation of the size exclusion. So this is will exclude proteins about 50 kilodaltons, 50,000 molecular weight. Inside is a white powder. In the beaker I put 9 grams of the G50 powder, and I swelled the powder overnight using this tris buffer, 30 millimolar tris buffer, pH 8.0. You can see that the 9 grams has swollen up to a volume of about 90 milliliters, almost 10 milliliters per gram of solid. Now we're going to gently stir the gel. It's important not to stir this too vigorously. Don't mechanically stir this. That can break your gel particles up into very fine dust, which will impede your flow rate in the column. So this is making a slurry. I'm using a sawed-off pipette to transfer my slurry to the column. For gel filtration, I want a fairly tall gel bed. So I'm trying to mostly fill this column. And excess buffer will drip out the bottom. So here you can see the drips coming out the bottom. Gel bed is starting to settle a little bit. Gel bed is right here. I'll add a little more buffer so that it doesn't run dry at the top. So I'm gently adding a little buffer to the top. This looks great. You can see the gel bed is fully settled here. There's a little bit of buffer on the top. I've capped the bottom right now to keep it from going dry. I'm going to put a mixture of two molecules onto the column. Blue dextran is a very large molecule. It's a polysaccharide. Molecular weight about 1 million. Fluorescein, on the other hand, is a small molecule. Molecular weight about 350 grams per mole. I chose these two molecules because they're nicely colored, so I'll be easily able to see the separation visually as they go through the gel filtration. I've drained the buffer, so now there's no residual buffer on top of the gel, and I've made a mixture of a total of 0.8 milliliters, 400 microliters of the fluorescein, 400 microliters of the blue dextran. Nice green color. I'll separate these on my gel. For this size column, this is getting near the maximum I would put on the column. I'm using a Pasteur pipette to gently load my material onto the column without disturbing the gel bed. So here you can see the blue molecules, the large molecules, are moving faster through the gel. So I'll hope that I can get a complete separation between the blue and the yellow. Now the excess on the top of the gel is all soaked in. So I'm adding buffer very carefully to the top of my gel filtration column. I'll just add a fraction of a milliliter first to help get everything into the gel before I add a large volume of buffer. So now the separation has proceeded somewhat. I'm going to try to add a decently large volume this time of buffer on the top. About a milliliter would be great. If I do this well, then the buffer that I put on the top remains colorless because all the colored molecules have already gone into the gel. Alright, now you can start to see a little colorless layer on the top. That's perfect. All the colored molecules are in the G50 and they're starting to separate quite nicely. The yellow molecules are up here, the blue are down here. It's still got a little overlap. So I may have overloaded this column, but we'll see. I'm hoping that before the blue molecules elute that they'll actually be separated from the yellow, but it's going to be close. So out of the bottom now, blue liquid is dripping. You can see I do have a colorless layer starting to develop here. The fluorescein has been slowed down by the gel filtration. The blue dextran is coming through. So this technique is often used to separate a protein, which is large, from the small molecules that could be salts or could be a fluorescent tag that is unwanted. I've topped up with buffer on the top so that it never runs dry. The fluorescein is now eluting from the column. Fluorescein is fluorescent. I'm shining a little blue light, UV light on it, and you can see it glows green. The last little bits of fluorescein are emerging from the column now. Here's the fractionation. This first tube has nothing in it. So this volume, which looks to me about 2.5 milliliters, is called a void volume, where nothing comes out. But then the blue molecules, the blue dextran, came through the column swiftly. So this is about a milliliter. Some of the blue dextran tailed into the next fraction. This is low concentration. Then I have a nearly colorless fraction here, and then the fluorescein starts coming out here. Fluorescein stopped coming out right about here. So the total volume will be 2.5, 3.5, 4.5, 5, 6, 7, 8, 9, about 10 milliliters is the total volume, which is what I would expect for a column of this size. Often what you want is just this first fraction with the large molecules. If you're trying to desalt a protein or remove the unbound label from a protein, then this is the fraction that has value. These fractions would just be junk. The original mixture, you can see, glows nicely. And what I believe is isolated blue dextran from the column has no glow in the UV. That's a sign that I have successfully separated out all the fluorescein from the blue dextran. So the column, if I've done it well, comes out completely colorless. It's reusable. Sometimes microorganisms like to grow in the cephadex, so if you want to preserve your column for a long period, keep it in the fridge, certainly. And sometimes azide is added to the buffer to prevent growth of microorganisms. Here's tris buffer with 0.01% sodium azide, and this will prevent microorganisms from chewing on the dextran that's in the column if you're storing it over a long period of time.
Yeah. So that was about the chromatography from the gel filtration chromatography. Now comes ion exchange chromatography. In this, you have a positively charged binds to the negatively charged beads. So in this, instead of carbohydrate beads, you have negative charged beads, to which your positive charged protein will attach to it, and the negative charge will flow out of it. So that's the main difference. So there will be an ion exchange chromatography and a cation exchange chromatography. In the cation and anion exchange chromatography, the matrix carries a net positive charge which interacts with the negatively charged proteins. And in another case, matrix is negatively charged, it interacts with the positively charged proteins. So let's see how we do this technique.
Ion exchange chromatography is a chromatography process that allows the separation of ions and polar molecules based on their charge. As well as other forms of chromatography, in ion chromatography, there is a mobile phase, which generally consists of buffer solution. And there is a stationary phase which consists of a matrix that contains charged ionizable functional groups. This type of chromatography is subdivided into anion exchange chromatography and cation exchange chromatography. In anion exchange chromatography, negatively charged molecules are attracted to a positively charged solid support, which is usually beads that provide a positively charged surface, such as resin quaternary ammonium. In cation exchange chromatography, positively charged molecules are attracted to negatively charged solid support, which is commonly beads that provide negatively charged functional groups, such as resin methyl sulfate. Ion exchange chromatography is frequently used for the separation and purification of proteins. The basic process of this chromatography can be represented in 4 steps: conditioning or equilibration of the stationary phase, sample loading, washing, and elution of the retained molecules. The first step is the equilibration of the stationary phase to the desired start conditions. The pH and ionic strength of the sample buffer are selected to ensure that, when sample is loaded, proteins of interest bind to the medium, and as many impurities as possible do not bind. Therefore, the same buffer is used for the equilibration of the stationary phase. When equilibrium is reached, all stationary phase charged groups are bound with exchangeable counterions, such as chloride or sodium. when the column is equilibrated. The second step is sample application. Depending on the pH of their environment, proteins may carry a net positive charge, a net negative charge, or no charge. This net charge can be determined by comparing the pHi of each protein with the pH of the buffer. Proteins are large biomolecules, consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain specific to each amino acid. Depending on the pH of their environment, the amino group can be protonated and become positively charged. The carboxyl group can be deprotonated and become negatively charged. And the side chain of some amino acids can become positively or negatively charged due to the gain or loss of protons. So, the net charge on the molecule can be positive, neutral, or negative. The isoelectric point, or pHi, is the pH at which a protein carries neutral charge. At a pH below their isoelectric point, proteins carry a net positive charge. And at a pH above their isoelectric point, they carry a net negative charge. In this example, the pH of the buffer is equal 5, consequently, protein A has a net negative charge. And both protein B and C carry a net positive charge. In addition, protein C is more positively charged, since its pHi is higher than the pHi of protein B. If we apply cation exchange chromatography, protein that has a net positive charge, it will bind to the negative charge beads of the stationary phase. On the other hand, uncharged proteins, or those with a net negative charge, would not bind. They pass through the column at the same speed as the flow of buffer. When all the sample has been loaded, the next step is column washing. The column is washed with start buffer. To ensure that all non-binding proteins have passed through the column. When all the sample has been loaded and the column washed, conditions are altered in order to elute the bound proteins. Most frequently, proteins are eluted by increasing the ionic strength of the buffer, or, occasionally, by changing the pH. As ionic strength increases, the salt ions compete with the bound components for charges on the surface of the medium, and one or more of the bound species begin to elute and move down the column. The proteins with the lowest net charge at the selected pH will be the first ones eluted from the column, as ionic strength increases. Similarly, the proteins with the highest charge will be most strongly retained, and will be eluted last. Protein B has the lowest net positive charge, so it can be eluted first. Then, protein C with the highest net positive charge, it can be eluted next, by increasing the salt concentration. Also, varying the pH of the buffer can be used to effect a separation, and to elute the retained proteins. As we seen, the pHi of the protein B is equal 7, and the pHi of protein C is equal 9. So if we add a buffer with pH above the isoelectric point of protein B, and below the isoelectric point of protein C, the protein B will become negatively charged, and can then be eluted. The protein C is still charged positively, consequently, still retained. Next, protein C can be eluted if we apply a buffer with pH above its isoelectric point. So, it becomes negatively charged, and it will pass through the column. One of the primary advantages for the use of ion chromatography is only one interaction involved during the separation. Another advantage of ion exchange is the predictability of elution patterns. By controlling changes in ionic strength or in pH of the buffer, using different forms of gradient, proteins are eluted differently in a purified, concentrated form.
Okay. So that was about that. Now there's affinity chromatography. So let's take a 5 minutes break here. Yeah, then we continue with further. lecture. Let's take a break just to have a mindset again to get for another chapter. So let's meet around 28 or 38 or 37. Yeah. And continue with affinity chromatography. So in affinity, बच्चे, let's continue. So this method relies on the knowledge of the properties of enzyme of proteins actually. So their antibodies must be available that have been raised against the enzyme, and the enzyme must be known to bind to some substrate. Such a ligand non-covalently in a reversible way. So it is the most specific chromatographic method because it relies on the specific binding of the target protein with its own specific ligand. This is your affinity chromatography. The use of glucose reagents to isolate glucose binding protein, conclavin A. So glucose binding proteins attaches to glucose residues on beads. So here we can see you load your buffer in pH buffer 7. You will have a protein not recognized by the antibody. The blue ones and the red ones will be, so they will get attached to it, and then you will have these blue and red ones separated. So let's check how it is done.
Separation in affinity chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and dilution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. Non-binding molecules will pass through in the flow-through during washing with binding buffer. In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by change in the buffer composition. A common way is to increase pH of the buffer.
Now next is HPLC, which is known as high pressure liquid chromatography, which is an enhanced method of a column method. In this, resin sizes are more refined, and all the chromatographic methods described above can be applied for the HPLC columns. So the reason for this is that HPLC columns are packed with much more smaller beads than those in the standard columns. The column is a large tube containing small particles known as stationary phase, and chromatographic bed is composed by the gel beads along with their inside columns. So once in the column, the sample mixture separates as a result of different components. So absorbance at 220 nanometer. So you have with different times, 5 or 10 minutes. So 1, 2, 3, 4, 5. So you can separate in these different peaks, different components actually. So now let's check how this HPLC machine works.
HPLC stands for High Performance Liquid Chromatography, but could equally well stand for High Pressure Liquid Chromatography. It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, the stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates.
How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase nonpolar. In paper chromatography, the solvent moves along the paper by capillary action. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high-pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, a component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a nonpolar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
Okay, so that was a little bit about HPLC. Then there is a FPLC, which is a fast performance liquid chromatography. It's a type of liquid chromatography where solvent velocity is controlled by the pumps. The pumps control the constant flow rate of solvents, and the solvents are assessed through the tubing from an outside reservoir. So I think so we're done with most of the good techniques that are required to separate, to isolate your protein, to quantify your protein, to purify your protein via chromatographic techniques. Yeah. So almost we are done with today's course here. There's nothing much to add to that. So if you have any questions, you are open for the questions now. Anything? Um, yeah. So now we can have some time for MCQs here. So answer these 5 answers to these 5 questions. This is A. So hurry up, student. Answer 1, 2, 3, 4, 5. What will be the answer of them? Hurry up, student. What will be the answer of these 5 questions? In large scale, I can show you how the large scale production is done of enzymes. I can share you one video about that later, but we are actually almost done for today. Our workshop is almost ends. So I just wanted to have few quick MCQs with all of you before we go ahead. So answer these 5 questions that I'm asking in the chat box. Yes, first answer is right, is all of the above. Second is protein, right. Third, yes, Sumner. Fourth, yes, it's A. And the fifth answer is 6, it's D. Rest of the students, please answer. Okay, now next 5. So you maybe try to answer these 3 first, then we go to the next. Hurry up, students. Okay, what would be the answer of competitive inhibitors of enzymes are? Majority of enzymes are active at SI unit of enzyme activity is? We have just discussed them today. Answer these three questions, बच्चे.